understand autoimmune disorders. And under autoimmune disorders, let's try to get the basics of autoimmunity first. So wait a minute. Yeah. So let's try to understand basics. The basic of autoimmunity, we understand what is self-antigen. We understand what is immature T cells. We already understand passive selection, negative selection. Basically, positive selection is that the TCR cells, right, test the. It's like a passive selection is like checking if your cells, if your precursor cells that are CD4 negative and CD8 negative, these cells, what happens is that these CD negative 4 these CD4 negative and CD8 negative, they get rearranged by the TCR genes, right, and they become CD4 positive and CD8 positive, and later they get tested for self antigen. Self antigen is just a piece of it's like a normal body component that the immune system targets, right, that immune system could target. So it tests, and what happens is that if your T cells, if your CD4 CD8 cells, they show a weak binding, it means they are good, they are healthy, they are not autoimmune. If they show a very strong binding to the self antigen, so under negative selection they get deleted, they get they perform what's called apoptosis, right, they die. So that's the basic concept of autoimmunity. Before going that, let's try to understand what is hypersensitivity. What's the concept of 4 type of hypersensitivity. Let's try to understand this. So hypersensitivity is that when we are sensitive to some sort of antigen that can be exogenous or endogenous, right? It can be antigen from within ourselves, or it can be an antigen that can be presented from the environment, right? And we have like thousands of antigens from outside, right? So what happens is that this antigen, this is like the thing that can trigger our immune system, right? That's the antigen. And the effector mechanism is that the part of immune system that is overreacting, right? Overacting, right? To that antigen, to that antigen. So we have to ask 2 questions. First of all, is it an antibody mediated, right? Antibody mediated means gamma globulin mediated, right? If it's an antibody mediated, or if it's a cell mediated, okay, that's very important. Cell mediated means neutrophil and blah blah blah. It's if this hypersensitivity, is it acute or is it chronic? That's very important as well. We can totally divide these hypersensitivity in 4 types, especially in 5 types. We'll talk. First is allergy, right? And this is a very acute form. What happens is that you get a first exposure, the antigen makes something called as IgE, that is gamma globulin E, okay, IgE. Now what happens is that this IgE, it coats means it surrounds the mast cell. These mast cells and basophil, they something called as, they do antigen cross-linking. Now what happens is that, for example, this is a mast cell, right? These are its hand. Now it will go to this antigen and it will cross-link this something called as IgE with each other, right? And this cross-links happens, and then we have something called as histamine and leukotrienes shower, right? Now mast cells, they produce a lot of histamine. Basophils, they also produce histamine and LK, leukotrienes, right? And once we have histamine and leukotriene, the effects will be itching, bronchospasm, and vasodilation. I hope we understand the basics of these effects. Now this thing, histamine and leukotrienes, they do this, right? The effect is this. Now what happens is there is presence of something called as anaphylaxis, eczema, rhinitis, right? There is presence, it means it happens in this this is the condition, right? These are like examples of IgE mediated allergy, right? Now what are these? Anaphylaxis, eczema, rhinitis. Type 1 is acute, like 1 second, okay? This is fast type, okay? So this is allergy. The other thing is antibody against cell surface. Cell surface, whose cell surface? Our own cell surface. Antibodies against our own cell surface. So it's a cytotoxic, right? It's toxic to our own cells. So antibody to the cell surface. What happens is that antibody binds to the antigen, right? This is an antigen, antibodies bind to it, right, on the cell membrane, right? This is a cell membrane or the extracellular matrix, right? Cell membrane of what? Cell membrane of our cells. Okay. What happens here is that these cytotoxic mediated attack is happening, right? The antibodies are attacking our own cells, our own cell membrane, our own extracellular matrix, right? Now, after this, what happens is that complement system activates. We just in previous video we understood what is complement system. Other thing, this ADCC system starts. What is ADCC? It's a natural killer cell system, right? Other thing is the blocking and the activating receptors. Now, let's try to understand it a little bit better. What happens here? This blocking and activating receptor. It means that. So what happens really here is that these antibodies, they bind to the receptors on our cell membrane, right? Our cell membrane, and depending upon what they do after binding, they can act in two opposites way. Okay, first is blocking and other one is activating. That's what I'm trying to understand myself. So blocking antibodies, it means that these antibodies, they block the normal ligand like hormones or neurotransmitters or whatever they have receptors for, right? These antibodies, and they sit on these receptors and they block the hormone that is supposed to meet on this membrane, right? These antibodies, they block the normal ligand from binding to its receptor. They inhibit receptor function, right? They block it. So where, wow, how? So in myasthenia gravis, right? This myasthenia gravis, antibodies against acetylcholine receptor, right? They block the acetylcholine receptors, right? So these antibodies, they sit on the receptor for acetylcholine, and what happens after this is no stimulus. So we know acetylcholine is an excitatory neurotransmitter, and then no stimulus is going to excite the muscle. So that's why we have a very weakened muscle tone in myasthenia gravis. Okay, that's one way. Other way of activating receptor is that these antibodies will mimic the natural ligand, right? The natural ligand that is supposed to sit on the receptor. They will activate the receptor without the real ligand being there. One of the cool shit about this is this cool thing about activating receptor is grave disease, not myasthenia gravis. Okay, I think I wrote it wrong. Okay, wait, let me correct it. Myasthenia gravis. Okay, so this is done. Myasthenia gravis shit is happening where the receptors are blocked. But grave disease, in grave disease, what happens is the antibodies, they bind to the TSH receptor on the thyroid cell, right? On the follicular cells. And on the follicular cells, when the TSH like antibodies bind to the thyroid, it activates it, right? And then thyroid production starts, and then we have a lot of T3 and T4, and then we have something called a T3 storm, right? We have this storm happening in our body, right? So it leads to hyperthyroidism. It leads to the breakdown of a lot of cholesterol, and it leads to a lot of less synthesis of steroid hormones, right? Because we have less cholesterol because we have hyper this T3, right? So this is what happens. So this is cytotoxic, right? If I'm saying it's cytotoxic, it means some antibodies, they just don't block it, right? They just activate it as well, like. And what happens is that cytotoxic means they kill the target cell through the cytotoxic mechanisms, right? So one of the mechanisms, we know it's a complement system, right? Complement mediated cytotoxicity. That complement mediated cytotoxicity, it's a type 2 hypersensitivity. Okay, type 2 hypersensitivity. Okay, now we understand this. Other thing that can happen is that hemolytic anemia can happen. Hemolytic anemia, it means that antibodies bind to RBCs, right? Here, hemolytic anemia, and once they bind to the RBC, the complement system destroys its own RBC. Other thing that can happen is ADCC. This thing, what is ADCC? ADCC is, let's try to open it up. Antibody ADCC, antibody dependent dependent cellular cytotoxicity. Okay, so ADCC, right? Now this what happens in this? What happens is that we have some cells. Those cells are called natural killer cells, right? And these natural killer cells, these NK cells, right? NK cells. These NK cells, they recognize the FC portion, right, of this antibody, okay? And then they release something called as perforins and granzymes, right? Now, what this perforins and granzymes do is that they poke these holes inside this cell membrane, and then these enzymes, they go inside and they perform the apoptosis of the cell, the death of the cell. Not apoptosis, it's like a cidal, it's like an attack, right? It's a vicious attack. Okay, so that is, so we understand complement activation, we understand ADCC, we understand the blocking mechanism, the activating receptor mechanism. Activating is for grave disease. Myasthenia gravis is for blocking. RH fever is also activating receptor, because just because how it's like this, there is a protein, it's called M protein, and this actually mimics our own own cardiogenic cells, right? And that's how it's able to like mimic our own cells, and that's how we have this something RH fever, and then we have this this whole RH thing happens, right? Because it can mimic, right? Now, when we talk about this thing called good pasture, we are really trying to connect the dots between this good pasture syndrome. What happens? Okay, so good pasture is like type 2 hypersensitivity again, right? Type 2, right? Again, what happens is that antibodies are made for your glomerular system, okay? Antibodies are anti-glomerular basement membrane, right? These are anti-GBM antibodies, right? Now they attack the kidney, they attack the glomerular basement membrane, right? The basement membrane that filters everything, and they attack alveolar basement membrane, right? That's very important. So once these antibodies, they go and they block it, and then the attack starts. The complement system attacks its own body, its own kidney, right? And then that's the system activation system start, and classic complement system start, and the formation of MAC, cell and membrane damage happens. And after this, the recruitment of inflammatory cells happens, basically C5a and C3a. They attract a lot of neutrophils and macrophages, and they release a lot of proteases and oxygen species and reactive oxygen species, right? So this everything, it results in damage of membrane, okay, and damage of capillary walls. So in kidney, necrosis of the basement membrane happens, right? And in lungs, the capillary destruction happen, right? Once the capillaries are destroyed, you will have something called as hemorrhage, right? You will have cuff blood in this good pasture, and you will have hematuria, proteinuria, because your membrane, the glomerular membrane, basement membrane is messed up in good pasture syndrome. Okay, I hope I'm able to explain it very simply. Let's try to understand immune complex mediated hypersensitivity. Okay, so what happens here is that the antibody complex is formed, and it is present in the serum, right? And what happens here is that here these antibodies, right, this complex antigen antibody complex, they deposit at the tissue, right? And once they start depositing in the tissue, right, what happens is that the complement system and the neutrophil system, they start, basically the inflammation and the complement system start, right? And again, the cool thing about this, that it is IgA, okay, plus antigen, right? I hope I'm able to, okay, antibody is IgA, okay, and this happens, right? Soluble antigen, right? IgA, immune complex deposition in the tissue, right? And it's like, it takes like hour to days. It can be like from 2 hours to like 2 days, okay? And what happens is that we have something called a serum sickness, right? And it's one of the reasons of SLE, right? And so that's the that's the concept. So we can see this thing happening in SLE, we can see this happening in foreign proteins, right? and post-strep glomerulonephritis. Okay, that's also true. So that's the story, right? It's the type 3 immune, right? Immune complex mediated. Three things clumped together: antigen plus IgA antibody plus the complement system, right? Type 2, we understand that it's antibody against the cell surface. It's fixed to a cell, so two sides of a cell membrane. I hope that's how I like remember. And type 1 is acute, so 1 second, that's how I remember. Okay, the fourth part is delayed T cell. It's like delayed, it means 24 to 72 hours, right? It's a cell. Now this is a T cell mediated. It's a delayed type. It's a cell mediated, right? And what happens is that T cells, we have two types of T cells, CD4 plus positive and CD8 positive, right? And there is no antibody, no type of immunoglobulin here, right? So we have Th1 cytokines, right? And then we have CD8. So CD8, right away, makes a lot of macrophages. And Th1, we have this. Listen, we have Th1 and we have Th2. Th2 leads to IL-10 and Th1 leads to IL-4 and IL-5, and IL-4 leads to these class switching, and IL-5 leads to class switching as well. So that's the concept of this whole thing, right? And we can see this happening where? We can see this happening here at like 2 to 3 days, and we can see this happening in dermatitis, in TB skin test, and in type 1 diabetes, right? And how? Type 1 diabetes, because there's a beta cell destruction in pancreas, right? In the islets of Langerhans, right? We know that in the whole beta cell tail of the pancreas, we have 4 types of cells: alpha, beta, I think gamma, and F cells or delta, like that. So in beta cells, they make peptide C, they make amylin, and they make insulin. So we have beta cell destruction happening because of this CD8 activity and Th1 activity, right? So that's the concept about hypersensitivity, right? So this is type 1, this is type 2, antibody against cell, this is type 3, type 4. Type 5 is this, blocking and activating receptors, right? So these are type 1, type 2, type 3, type 4, and type 5. This is the classical type of hypersensitivity.